Controlling microorganisms involves measures to inhibit the growth, eliminate, or prevent the spread of harmful microorganisms. This is critical in healthcare, food safety, and public health. Microbial control methods can be divided into physical, chemical, and biological approaches, but in this class, we are going to study the physical and chemical approaches of microbial control methods. What is sterilization? Sterilization is the killing of all microorganisms in a material or on the surface of an object, making it safe to handle without fear of contamination. Any material that has been subjected to this process is said to be sterile. Although most sterilization is performed with a physical agent such as heat, a few chemicals called sterilant can be classified as sterilizing agents due to their ability to destroy spores. A germicide or microbicide is any chemical agent that kills pathogenic microbes. Germicide can be applied on inanimate materials or on living tissue, but it ordinarily cannot kill resistant microbial cells. Any physical or chemical agents that kill germs have germicidal properties. Disinfection is the use of chemical agents or disinfectants that destroys or removes all pathogenic organisms. This disinfection destroys vegetative pathogens but not bacterial endospores. It is important to note that disinfectants are normally used only on inanimate objects because they can be toxic to human and other animal tissues when used in higher concentration. Disinfection also removes harmful products of microorganisms, for example, toxins from materials. Examples of disinfectants is applying 5% solution of bleach or sodium hypochlorite on tables, boiling food utensils, immersing thermometers in an isopropyl alcohol or ethanol alcohol solution between use, using hydrogen peroxide, and using iodophores. Antiseptics are chemical agents applied directly to the exposed body surfaces or living tissues like skin, mucous membranes, and wounds. These are used to destroy or inhibit vegetative pathogens. Examples of antiseptics is preparing the skin for surgical incisions with iodine compound, swapping open root canal with hydrogen peroxide, washing hands with germicidal soaps, alcohol like isopropyl alcohol or ethanol alcohol, and phenol. Sanitization is any cleansing technique that mechanically removes microorganisms along with food debris to reduce the level of contaminants. Sanitizer is a compound like soap or detergent with alcohols that is used for sanitization. Sanitizer used to sanitize cooking utensils, dishes, bottles, cans, and clothes. Sanitizer may not make completely free of microbes, but make materials safe for normal use. Air sanitization with ultraviolet lamps reduce airborne microbes in hospital rooms, veterinary clinics, and laboratory installations. Preservation is a general term for measures taken to prevent microbe-caused spoilage of susceptible products, that is, pharmaceutics and foods. Decontamination is the removal or count reduction of microorganisms contaminating an object. In this case, the objective of aseptic measures and techniques are used to prevent microbial contamination of materials or wounds. In aseptic measures, Chemical agents are used to fight pathogens in or on living tissues. Let us see physical methods of sterilization and disinfection that involve heat. Application of heat is simple, cheap, and effective methods of killing pathogens. The first physical method is pasteurization. This is antimicrobial treatment used for foods in liquid state. This pasteurization can be low temperature pasteurization, high temperature pasteurization, and vaporization or ultra high temperature pasteurization. In case of low temperature pasteurization, 61 degrees Celsius for 30 minutes and 71 degrees Celsius for 15 seconds must be applied. In case of high temperature pasteurization, a brief seconds, that's about 15 to 20 second exposure of materials to 80 to 85 degrees Celsius in continuous operation. In vaporization or ultra-high temperature pasteurization, heating to 150 degrees Celsius for 2 to 5 seconds in a pressurized container using steam injection. Since pasteurization apply temperatures below what would be required for sterilization, less effective and does not kill bacterial spores. The second physical method of sterilization is dry heat sterilization. This includes the first one, flaming. This flaming includes sterilization of inoculating loops or wires, tip of forceps, and searing spatula. This flaming is carried out by holding material in a flame of Bunsen burner until the material becomes red hot. The second dry heat sterilization is incineration. This is an excellent method for safely destroying ineffective materials by burning them to ashes. It has many uses. 1. Incinerators are used to carry out this process and are regularly employed in hospitals, research labs to destroy hospital and laboratory wastes. The method is used for complete disposal and destruction of infectious materials such as syringes, needles, culture materials, dressings, bandages, bedding materials, animal carcasses, and pathology samples. This method is fast and effective for most hospital wastes, but not for metals and heat-resistant glass materials. The third forms of dry heat sterilization is hot air oven. This requires exposure to 160 to 180 degrees Celsius for 2 hours to 30 minutes, which ensures thorough heating of objects and destruction of spores. Another physical method of sterilization and disinfection is moist heat sterilization. This involves autoclave. This is a charged with saturated pressurized steam. This involves 121 degrees Celsius for 15 to 20 minutes with 1 atmosphere of pressure, or it involves 134 degrees Celsius for 3 to 5 minutes with 2 atmosphere of pressure. Another forms of moist heat sterilization is intermittent sterilization. Certain heat level substances, for example, serum, sugar, and egg, etc., that cannot withstand a high temperature of the autoclave can be sterilized by a process of intermittent sterilization or tendalization. Tendalization is a technique involving repeated cycle of heating and cooling for several days. This tendalization is carried out over a period of 3 days and requires a chamber to hold the materials and a reservoir for boiling water. Items to be sterilized are kept in the chamber and are exposed to free-flowing steam of 100 degrees Celsius for 20 minutes for each of 3 consecutive days. Day 1, Heat the material at 100 degrees Celsius, that is steam or boiling water, for 20 to 30 minutes. Then allowing materials to cool and incubate at room temperature overnight. Day 2, repeat heating to kill newly germinated vegetative cells, then cool and incubate again. At the third day, repeat the heating to kill newly germinated vegetative cells. So this day, ensure complete sterilization.